Okay, so I'm going to talk a little bit about this grant, which is in the same funding cycle as the feed efficiency grant that you're familiar with, also focusing on a hard-to-measure trait, which is bovine respiratory disease. And go through a little bit of the research progress to date and some of the different studies that are involved in this particular grant. I should say I'm the extension lead on this grant. It's obviously a multi-institutional effort, and Holly Nyberg is actually from Washington State University, is actually the research lead, and so I'll be presenting some of the data that's coming out of that research effort. Just to give you an idea, and I probably don't need to tell anyone here that bovine respiratory disease is a big problem, but it really is actually a mind-boggling problem in terms of the number of animals that die from this disease every year, and the losses to the beef cattle industry and the cattle industry in general on an annual basis is in excess of $1 billion a year. It's not only beef, it's also dairy cattle that get it, although in a somewhat different time frame just given how they're raised. But it's responsible for a large number of the mortalities in both unweaned and weaned dairy heifers, and then also non-predator losses in the beef cattle industry. You're probably familiar with the fact it's a multifactorial disease. That's why it's called the complex. It's a bovine respiratory disease complex associated with many bacterial and viral pathogens, and it tends to be exacerbated in times of stress when the immune system of the animal is low, consequently often called shipping fever, because animals are under stress during times of shipping. And this number kind of shows a sad tale to me in terms of what we've done to decrease the incidence of animals actually dying in feedlots. We're up at almost 1% level there of animals entering feedlots that are dying from this disease. And although there's been improved vaccine developments and a number of super antibiotics that have come out over the years, there really hasn't been a sustainable decrease in this particular incidence of disease. And the next talk is going to speak about the sustainability of the beef industry, and having 1 in 100 drop dead in the feedlot is not going to help the sustainability of the beef industry. And so it really is a problem that is looking for a solution, a different solution to the current approach of vaccine management. And disease resistance is a very attractive target trait, not unlike feed efficiency, in the fact that it's a very valuable trait. It is also coupled with increased concern about the use of antibiotics in agriculture in general, animal agriculture in general. Anything we can do to improve the genetics to reduce the incidence of disease is going to be both better for the animals, better for the industry, and also have the knock-on benefits of reduced antibiotic use. And it really, as geneticists, of course, we would always say that, but anything you can do genetically is permanent and long-term, and it's going to have a sustained benefit to the industry. And other animal industries have been able to successfully target disease. Even though disease, susceptibility, resistance tends to be a low heritability trait, it is a trait that other industries, specifically the dairy industry with regards to mastitis, and the poultry industry with regards to Merrick's disease and other diseases, have been able to put selection emphasis on while still maintaining productivity traits and other traits of interest to their particular industries. And so there is the potential to make genetic improvements in these type of traits despite the relatively low heritability. So at the time that the feed efficiency grants were announced in April 15th of 2011, there was also the bovine respiratory disease complex grant announced that is actually a $10 million 5-year grant with Texas A&M being the lead institution. And the overall long-term goal of the BRD cap is to reduce the incidence of BRD in both beef and dairy cattle by capitalizing on recent advances in genomics, to enable genetic approaches to select for cattle that are less susceptible to disease. I don't use the term resistance because resistance to me is kind of a categorical thing. If you're resistant, you're never going to get sick. We're not going to make animals that are bulletproof. What we're trying to do is decrease susceptibility. So select for animals that are less likely to get sick, but if you still don't wean them appropriately and background them appropriately, they will still get sick. This is both an environmental and a genetic issue, and both of those have to be handled together in order to have a sustained effect. And so resistance is a little bit of a strong term, although people sometimes use that somewhat interchangeably. There are a number of universities involved in this project. I'm standing here as a representative of the CAP team. It's led by Jim Womack out of Texas A&M. As I mentioned earlier, the research lead is Holly Nybergs out of Washington State University. Milt Thomas out of Colorado State is the educational lead, and I'm the extension lead out of UC Davis. And similar to the feed efficiency, we also have the research, the education, and the extension simultaneous goals associated with that particular. So if we look, we're spread throughout the nation and have presences in areas that have very high cattle concentration, particularly dairy in California, and of course Texas is a very big beef state. The research team is all listed here. We obviously weren't selected on our phenotypes, but there is, if you're interested in learning more about the project, you may remember last year we had a group from Cattleman to Cattleman, NCBA's Cattleman to Cattleman's program that was filming here at the Meat Animal Research Centre, and that went to air Tuesday, October 16th of last year. And if you're interested in having a look at that, I have a tiny URL.com there that's just called Cattleman to Cattleman, CTC BRD CAP, and if you just click that in, you can have a look, and it's about a 10-minute program that kind of goes through what we're trying to achieve with this particular grant. But what we're interested in doing is trying to develop some type of DNA tests for BRD susceptibility. And as has been alluded to earlier by other speakers this afternoon, what do you need in order to accomplish such a goal? You basically need large training or discovery populations. What we've talked about to date has been utilizing breed association resources where we have large training populations, where we have phenotypes or EPDs on traits that are already incorporated in national cattle evaluation. What we're talking about here is a trait that currently isn't in national cattle evaluation, and consequently there aren't large pre-existing populations for us to go and mine and develop DNA tests from. And so consequently we're going to have to go out and get these populations and phenotype them and genotype them ourselves, in the same way the Feed Efficiency Project is having to do that analogous thing. And we're also going to be working with phenotypes, not EPDs, and as was alluded to earlier by Matt, when you've got an EPD, you've got a lot of information there, a lot of phenotypes, whereas in this case, we're actually going to be taking individual animal phenotypes, and consequently we're going to need larger populations than the 1,000 or so that has sufficed in order to accomplish the similar goal with regards to traits that are already in national cattle evaluation. And as has been discussed before, and thank you everyone for leading in so I don't have to explain this, from that we would develop a prediction equation that can be used to predict the genetic merit of young animals, in this case for the phenotype of bovine respiratory disease susceptibility. So if we're going to have genotypes and phenotypes on animals that are sick with bovine respiratory disease, we're not going to go to university herds in order to accomplish that. We're going to need to go with commercial partners in order to get the numbers of animals that we're going to need in order to successfully complete this project. And consequently, we have a fairly tight working relationship with large commercial entities to do the genome-wide association study part of this particular project. We also have some work that is getting done on universities, I'll talk about later, and there I think it kind of contrasts the value of cooperating with large commercial entities to get large numbers of phenotypes for certain things versus doing things that no sensible commercial person would ever want you to do to their cattle, which is intentionally infecting the cattle and making them sick with bovine respiratory disease. And that's where the kind of the benefit of having university populations where you can actually do that type of experiment is really important. So our first research objective is, and really the meat and bones of the BRD CAP, is to identify genomic regions associated with bovine respiratory disease. I'll use the term susceptibility in both beef and dairy cattle. And so the overall arching goal here is try to develop data sets, 3,000 in beef, 3,000 in dairy, where we basically have a case control design. So an animal that's sick with bovine respiratory disease, that's a case, and a parallel control that's matched up to that animal that is not sick with bovine respiratory disease. And I'll go through a little bit where we got these populations, where basically our discovery is going to be 2,000 animals in dairy, 2,000 in beef. that are matched up case control, so 1,000 case, 1,000 control. And then we have a separate independent population in both of those industries where we'll be doing 500 case, 500 control. That will be where we'll validate or determine how well our predictions are working in terms of predicting animals that we think should get sick versus won't get sick. And we're going to be genotyping all of those 6,000 animals on the 770 high-density SNP chip. And so I'll talk a little bit about the importance of accurate diagnosis. And one of the reasons that heritability of disease, resistance, susceptibility is so low is there's a lot of different factors that play into whether an animal is sick or not. It depends whether they've been exposed to the pathogen, how healthy they are, what good nutrition they have, and also whether you're very good at picking whether an animal is actually sick or not. And in the case of feedlots, we've typically used this depression, appetite, respiratory rate, temperature elevation as a guide, or the DART guide, for picking out those animals that are sick. And a good pen rider can pick out sick animals, but it's also possible for an animal to mask its sickness and not get picked, and another animal that isn't sick to get picked. And you can imagine in a case control design, it's really important to make sure the ones you say are sick really were sick and the ones that were healthy really were healthy, or you're going to confound your data and you're going to have a much harder time pulling out the genetic signals that are uniquely associated with the animals that are sick or healthy, whichever way you want to look at it. And in some cases, and this is a sad little calf that's on one of our studies, I think even I probably could pick that this animal isn't feeling too well, and I think I might say that that one is definitely symptomatic of having bovine respiratory disease, and that's a very easy one to pick. But what we'd like to get, and actually I think his penmate has the same problem, what we'd like to get is a fairly objective approach to classifying animals, whether they're sick or not. So the symptomatic animals, typically in the typical DART system, temperature plays a really important role, and if the rectal temperature is above about 103 degrees Fahrenheit, that is used often as a key to say that that animal's sick. So it looks depressed, it gets brought in, they take a temperature, if it's above 103, typically it'll get treated with some sort of an antibiotic. I'm going to show you some data here about the dangers of using temperature as your sole guide for determining bovine respiratory disease, especially in climates like outside today, I dare say, or in hot climates like California in the summer, because you really can't, that's a really not on its own isn't sufficient to determine whether an animal's sick or not. And so there's a lot of confounding factors that determine whether you get animals correctly classified as sick or not. And part of it is really, the diligence and astuteness of those checking the animals, and how well those animals are actually displaying that disease and the disposition of the animals. And so what we tried to do in this project was have a very objective approach to classifying our cases and controls. And what we basically did in the dairy study was use this system that's been developed by Sheila McGurk at the University of Wisconsin, where it's basically a scoring criteria of a number of different parameters. And the things that we took into account were the temperature of the animal. So if it's greater than 103, then it scores a 3 on this particular scale. If it's in this range, a 2, and a normal healthy animal would get 0. So a normal healthy animal would have a normal rectal temperature, no cough, no nasal discharge, normal eye scores and normal ear scores, versus a very sick animal over here would get a score of 3 for temperature, 3 for coughing, so now it's 6, 9 for snotty nose, another 3 for ocular discharge, and then this bilateral head droop would add it up to 15 total. And these are some kind of calves that we picked in the dairy study that you can see would be classic case calves because they've got a number of these different symptoms. And using this objective approach, we were able to classify based on a numerical system whether or not an animal was sufficiently sick and exhibited all these signs to get classified as a case. And then what we tried to do is select an animal very close by in the raising system that was not exhibiting any of those symptoms to try to minimize as much as possible the environmental variability between the animals and have them have had similar experiences and exposures, one's sick, one's healthy, and that way try to effectively increase the heritability because we've minimized the environmental variation and let the genetics come to the fore. And so this project was done, and this has actually been completed on the dairy population, and that's the beauty of having these large commercial populations. This is a dairy calf raising ranch in California that has a 70,000 head capacity. And although this may look like a chicken coop, it's actually little dairy calves here, and I think there's maybe 300 per row or something like that, and there's one here, there's one here, there's one here, there's one here, and you can get the idea. And so in the morning you could walk along and get a lot of different observations and really pick out those ones that classify into our sick criteria, and maybe all of this lot, maybe all come from the same dairy on one particular day, and every day you'll get a few thousand in. And it's a very large scale project, but it's perfect for the type of research that we were trying to do. So we were basically able to use 1,000 case and 1,000 control 30 to 60 day old dairy calves. That's typically when these calves get sick with bovine respiratory disease. We used that respiratory scoring chart to objectively classify cases and controls. Then we took blood for DNA, and then another thing we did was a deep pharyngeal nasal swab, and then used that swab to identify any viruses that were present in that animal and the classical bacteria that are associated with bovine respiratory disease. And we went to a great deal of effort to get the deep pharyngeal nasal swab, which basically involves passing a fairly long swab down into the pharyngeal cavity to try to avoid just getting commensal bacteria that are just hanging out in the nostril, and really get any bacteria and viruses that are down in the lungs. It's theoretically less stressful to the animal than a lung lavage, although I think some of the calves might disagree with that because they didn't exactly enjoy having their deep pharyngeal nasal swab done. And the next slide is kind of an interesting slide. Only a vet student would think to do this, but they basically drilled the head in half here, and you can see this is our pharyngeal swab going down. So that if you just did a nostril swab, you'd get this bit, but where we're going down here is into the deep nasal pharynx to try to it's a guarded swab, so it only picks up the bacteria and viruses that are kind of back here, to try to really minimize ones that are just hanging out in the environment and get the ones that are involved in the lung issue that's going on. And like I said, it's meant to be less stressful than a bronchoalveolar lavage, but I'm not sure all the calves agreed. So with the cases, we tried to score in exactly the same way, and we would try and pick one in an adjacent hutch that came from the same dairy and sex. And needless to say, they were all the same breed. They were Holsteins. And so that, of course, is going to simplify our life in terms of, well, hopefully, in terms of looking for SNPs that might be associated with bovine respiratory disease. And then all of this data was collected from those similarly to the cases. And really, the ready availability of the SNP chips that was discussed earlier today is really what's enabled us to even conceptualize doing a project of this magnitude. And the hope is that the high-density SNP chip is going to be dense enough to be in close association with actual causative SNPs that are associated with improved susceptibility to bovine respiratory disease. So basically where we are with this, we've collected all of the samples on those 2,000 calves. We've done the diagnostics for the different bacteria, and we've DNA extracted and genotyped all of those 2,000 animals. The Bluppy guys, the quantitative geneticists have been playing with their different programs to have a look at where we're seeing significant associations in the genome, using various different independent approaches and having a look at how much overlap they're getting in terms of areas of the genome that look like they're really associated with animals that are sick versus healthy. And this is data that's just really unpublished at the moment, but we are getting regions of the genome, depending which analysis you use. There are some regions that are common to all the different analyses, which gives you a little bit more confidence that we might actually be getting something serious here. It's different regions of the genome where we're seeing SNPs that are in association with animals that are sick or healthy. And really the bottom line is how much of the variation in the incidence of the various traits and overall the bottom line here, BRD, we're explaining about 20% of the variation in the incidence of BRD with the SNPs that have been isolated to date. Currently, we're in the process of validating those in the independent dairy cattle population. So that's all very easy when you've got 70,000 calves laid out in little chicken coops to get that done. It's going to be a little bit harder, of course, to get that done when we're talking about beef cattle in a feedlot situation. And that experiment is underway. It's being done at a cooperating feedlot in Colorado. We decided to focus just on Taurus cattle, trying not to get Indicus influence to try to keep the genetics as less diverse than you would get if you had eared cattle. And we've got about 900 of these collected so far, so 450 case and 450 control. And of course, bringing in a control animal in a commercial feedlot when it's healthy and doing all of this to it is a lot more to ask than just taking a swab from the animal standing next door in a hutch, and so it's a bit more of an ask on the beef cattle end of things. But the idea is to eventually get the same number of animals from beef cattle populations as well. So that's like I said, the meat and guts of the bovine respiratory disease cap. I'll just go through one of the other objectives because I think it's kind of an interesting one, and this is also actually in collaboration with some people here in the audience, and specifically Jerry Taylor. And what we're interested in here is looking at the transcription profiles of which genes get expressed when an animal gets exposed to the different pathogens associated with bovine respiratory disease. And UC Davis had the pleasure of actually infecting our healthy cattle with intentionally infecting them, not vaccinating them, and making them sick with bovine respiratory disease and then killing them. And so this is one that we had a bit of a hard time getting a commercial partner that thought that was a good idea. So the university herd was quite happy to do that, or not so happy, but it was basically was done at our facilities at Davis, and we took, you'll understand what this took, but we basically were working with about 700-pound Angus calves that were challenged. There was 6 in each group that were challenged with the different bacteria here that are associated with bovine respiratory disease, or the different viruses, and these were done individually, so not all of, so one got one bacteria, or 6 got Mycoplasma bovis, and at peak infection we then killed those animals, which doesn't make your facility manager very happy, and we took all of their different parts of their body to do RNA-seq analysis of various tissues, particularly with a particular emphasis on the type of lymph tissue that you would expect to be going crazy when you've just infected an animal with viruses and bacteria. And this is really being done with Dr. Laurel Gershwin, who's a veterinary doctor at the University of California. And so to infect them with with the bacteria, we basically grew up a bunch of bacteria and put them in this big syringe here, and then put a nose tube down into the animal's the cavity there, and then just basically injected those bacteria into the animal, and basically overwhelmed its ability to get rid of that particular bacteria. And similarly with the virus, we used what's called a nebulizer that's basically aerosolized the virus and just put it over the poor calf's mouth for about 10 minutes whilst it inhaled huge quantities of viral particles, and it of course hadn't had vaccinations for those viruses, and we were very successful at getting them very sick. So I guess that's good. But this is just a graph I thought was kind of interesting, is we had, in addition to all of the other insults we did to these calves, we had an indwelling rectal thermometer that took a temperature reading every 5 minutes. And basically what this shows here is these are our case calves are in are blue, and our control calves are red. And this is basically the day-to-day diurnal variation that occurs as the temperature goes up and down during the day. And I just drew kind of a black line here because this is 39.5 degrees Celsius, which is about 103 degrees Fahrenheit. And what you can see here is, and typically, you know, 103 or 100 in that ballpark is kind of the trigger when you bring a cow in, or a steer or an animal that's sick, and you do a rectal temperature. You say, oh, well, we better treat that one. It's got, you know, it's diseased. It needs antibiotics. But you can see here, this is the night. This is obviously the middle of, or probably late afternoon. You can have a look down here at the different, these are different days. So we infected our animals here with bovine respiratory syncytial virus, and you can see that the control group and the treated group are continuing on sync together. And then here at about day 3 or so of infection, you see the blue guys start to draw apart and become quite significantly warmer or hotter and infected and feverish compared to the controls. But I wanted to just draw your attention to this day. It just happened to be a particularly hot weekend in Davis that particular weekend. And basically none of the groups, either the controls or the sick ones, dropped below 103 the entire weekend. And so if you pulled one of those animals in and you took a temperature and 103 was your cutoff, I could pretty much tell you you would be treating them for bovine respiratory disease if you didn't also look at the other attributes of that animal. So those animals have all been infected, slaughtered, and we've chopped up all of the little bits of pieces of their different tissue. And basically what we took from those animals was lung lesions, if that was present in the lungs, and actually the viruses made those lesions come up very quickly if you did that. Different lymph tissues, bronchial nodes, liver, spleen and blood. And that's all now gone to the University of Missouri, where a postdoc with Jerry Taylor is in the process of having a look at what happens in comparison between the ones that were treated and the ones that and control animals, to see what genes are getting regulated and how they differ between the different exposures to viruses and bacteria, with the hope of working that back into the overarching objective, which is to try to put it all together. And okay, we see an area in the GWAS that's suggestive of something, and now maybe you see an upregulation of one particular gene in that area, and kind of marry it all together to give us the best possible targets that we could then put into a DNA test, which is the ultimate goal of this particular project. So obviously we're capitalizing on the advances in genomics. There's also educational and outreach objectives associated with this CAP, similar to the feed efficiency project. And you may notice there's a lot of these type of projects that are getting funded at the moment, and they're really focusing on these difficult to measure traits: feed efficiency, respiratory disease or diseases, because, you know, it's really not something that's that easy to do, get your phenotypes on, and we need these large resource populations in order to be successful at these types of projects. And there's the feed efficiency program that Matt and a lot of you are involved in here. There's the program I just mentioned here. There's a recently funded program led by Patterson out of Missouri that is focusing on fertility, another hard to improve trait, looking particularly for missing homozygotes, as was discussed earlier, and there's also one in dairy cattle on reproduction. And so all told, there's more than $20 million in active AFRI grants targeting these hard to measure traits. And I think the bovine community at the moment is in a really nice situation to capitalize on the information that's coming out of these genomics projects, with the hope that we'll actually be able to make genetic improvement in some of these really hard to measure traits. Having said that, I think, and this is my last slide, Matt, I think you know, one of the difficulties, and everyone in this room is well aware of the fact that if you look at the breeding pyramid associated with some industries, it is a pyramid. The beef industry has a little bit of a different structure, and we've kind of got little pyramids, I would argue, with the different breeds at the top of the breeding pyramid here. We have our large group of 30 million or so beef cows that are part of the commercial chain, and we have kind of the change of ownership quite often between these different sectors, coming into the stockers and backgrounders, and then finally down to the feedlots, where we've got relatively few operations, and then even fewer processes. And we're working often in traits. For example, BRD is something that's going to have a tremendous value to the feedlot industry. But somehow, I think for the technology to kind of take off here, there has to be some reward, if you want these little pyramids up here, if you will, to focus on traits that are going to be of importance to this sector down here. Similarly, feed efficiency, I think, is a trait of great value here, although you could argue it's also of great value to a cow-calf operation also. But there is really, you know, I think for it to really take off, there's going to be some sort of a value transfer back to the little pyramids at the top here to include these types of traits in their selection decisions in order for it to be taking off in the industry as a whole. So thank you very much for inviting me, and I'll finish there.